Welcome to today's podcast, and we are discussing an important question in neonatal nutrition. Does it matter whether nutrients are delivered continuously or as intermittent meals? This question is especially relevant for premature and medically fragile infants who cannot consume enough milk orally and therefore receive nutrition through a feeding tube. Tube feeding can generally be delivered in two ways. Nutrients can be given continuously at a slow and constant rate throughout the day, or intermittently as separate meals given every few hours. Both approaches can provide the same total amount of energy and protein. However, they may not produce the same metabolic response. A study published by El-Kadi and colleagues examined whether intermittent feeding produces a stronger anabolic response than continuous feeding in neonatal skeletal muscle. The study was titled, "Anabolic Signaling and Protein Deposition are Enhanced by Intermittent Compared with Continuous Feeding in Skeletal Muscle of Neonates." Let us examine what the researchers did, what they found, and why these findings matter. The neonatal period is characterized by extremely rapid growth. During this stage, skeletal muscle has a remarkable ability to use dietary amino acids for protein synthesis. This response is much stronger in neonates than in older animals or adults. After a meal, circulating concentrations of insulin and amino acids increase. These signals activate molecular pathways inside muscle cells that stimulate the translation of messenger RNA into new proteins. One of the most important pathways involved in this process is the mechanistic target of rapamycin, commonly called the mTOR pathway. The mTOR pathway acts somewhat like a nutrient sensing control center. When amino acids, particularly leucine and insulin, become available after feeding, mTOR activates several proteins involved in translation initiation. Translation initiation is the first major step in building a new protein. However, this anabolic response does not remain elevated indefinitely. In neonatal muscle, protein synthesis increases rapidly after a meal, but can return toward fasting levels within a few hours. This raises an important question. If nutrients are supplied continuously, insulin and amino acid concentrations may remain relatively constant. Could the muscle become less responsive to this constant nutrient exposure? In contrast, intermittent feeding produces repeated increases and decreases in nutrients and hormones. Each meal may generate a new anabolic signal. This was the central idea tested in the study. The researchers used neonatal pigs as a model of human neonatal metabolism. Neonatal pigs are commonly used in nutrition research because their gastrointestinal development, body composition, and metabolic responses to feeding share several characteristics with those of human infants. The pigs were approximately 9 days old at the time of the experiment. They were divided into 3 groups. The first group was fasted. The second group received nutrition continuously at a constant hourly rate. The third group received the same total amount of nutrition as intermittent meals delivered every 4 hours. Importantly, the continuously and intermittently fed pigs received the same total amount of food, protein, and energy. Therefore, the study was not testing whether one group received more protein than the other. It was testing whether the pattern of nutrient delivery changed the anabolic response. The feeding treatments continued for approximately 29 hours. The researchers also used stable isotope traces of phenylalanine and tyrosine. These traces allowed them to estimate protein synthesis, protein degradation, amino acid uptake, and net protein deposition across the hindquarters. Because a large portion of the tissue in the hindquarters is skeletal muscle, this approach provides important information about muscle protein metabolism. The researchers also collected muscle samples and measured proteins involved in anabolic signaling, including components of the mTOR-dependent translation initiation pathway. After an intermittent meal, arterial insulin concentrations increased. Concentrations of branched-chain amino acids, phenylalanine, and tyrosine also increased. These post-meal increases were greater in the intermittently fed pigs than in the continuously fed pigs. This is important because insulin and amino acids work together to stimulate muscle protein synthesis during the neonatal period. Continuous feeding provided nutrients throughout the experiment, but it did not produce the same pronounced rise in circulating insulin and amino acids. In other words, the continuously fed animals received the nutrients, but their muscles were not exposed to the same strong, repeated post-meal signals. Intermittent feeding increased the activation of proteins involved in translation initiation. One important finding was increased formation of the EIF4 E-EIF4G complex. This complex helps recruit messenger RNA to the ribosome, allowing the cell to begin translating the genetic message into a protein. The activation of this pathway indicated that intermittent feeding produced a stronger molecular signal for muscle protein synthesis than continuous feeding. This supports the concept that skeletal muscle does not respond only to the total amount of amino acids supplied. Muscle also responds to the magnitude and timing of changes in amino acid and insulin concentrations. A meal creates a clear transition from a relatively low nutrient condition to a high nutrient condition. That transition appears to be an important part of activating neonatal muscle protein synthesis. The most important result was that muscle protein synthesis increased after intermittent feeding. In contrast, protein synthesis remained relatively unchanged in the continuously fed and fasted groups during the corresponding measurement period. The intermittently fed pigs also demonstrated greater net amino acid removal across the hindquarters. Net amino acid removal means that more amino acids were taken up by the tissue than were released from it. This contributed to greater net protein deposition in the intermittently fed pigs. Therefore, intermittent feeding did more than temporarily activate a few signaling proteins. The molecular response was accompanied by a measurable increase in protein synthesis and protein deposition. Protein deposition represents the balance between two processes. Protein synthesis builds new proteins, while protein degradation breaks existing proteins down. The researchers originally considered the possibility that intermittent feeding might enhance protein deposition both by increasing synthesis and by decreasing degradation. However, protein degradation did not differ substantially between the feeding groups. The improvement in protein deposition with intermittent feeding was therefore driven primarily by an increase in protein synthesis, not by a major reduction in protein breakdown. This distinction is important. Intermittent feeding did not appear to preserve muscle simply by slowing protein loss. Instead, it actively increased the construction of new muscle protein. The study also measured proteins associated with the ubiquitin, proteasome, and autophagy lysosome systems, which are major pathways of intracellular protein degradation. Most markers of these degradation pathways were not substantially affected by the feeding pattern. One exception was LC32, which was highest in the fasted animals, although the authors cautioned that this marker alone does not provide a direct measurement of protein degradation. Continuous feeding supplies a steady stream of nutrients, but the anabolic machinery may not remain maximally activated during prolonged nutrient exposure. The muscle may initially respond to the nutrients, but the response can become attenuated even when amino acid concentrations remain elevated. This phenomenon is sometimes described as a muscle full effect or anabolic refractoriness. The muscle temporarily increases protein synthesis after nutrient availability rises, but it cannot remain fully stimulated indefinitely. With intermittent feeding, amino acid and insulin concentrations rise after each meal and then fall before the next meal. These repeated nutrient pulses may allow muscle to respond again when the next meal arrives. Therefore, intermittent feeding may create multiple distinct anabolic opportunities, while continuous feeding provides a flatter and potentially less effective signal. Not necessarily. This was a carefully controlled animal experiment designed to examine metabolic mechanisms. The pigs were studied for only about 29 hours, and there were 6 animals in each treatment group. A hospitalized premature infant may receive tube feeding for days, weeks, or even months. Clinical feeding decisions must also consider feeding tolerance, gastrointestinal function, blood glucose control, the risk of aspiration, medical stability, and the infant's ability to tolerate a bolus meal. Some infants may not tolerate intermittent meals and may require continuous feeding. Additionally, improved muscle protein synthesis during a short experimental period does not automatically mean that intermittent feeding will produce better long-term growth, neurological development, or clinical outcomes in every infant. The study provides strong mechanistic evidence, but it should not be interpreted as a universal clinical feeding recommendation. Longer-term studies are needed to determine whether the enhanced protein deposition observed with intermittent feeding persists over time and translates into clinically meaningful improvements. This study provides 3 major insights. First, providing the same amount of protein and energy does not guarantee the same metabolic response. The timing and pattern of nutrient delivery matter. Second, intermittent meals generated stronger increases in insulin and circulating amino acids than continuous feeding. These increases activated mTOR-dependent translation initiation signaling and stimulated muscle protein synthesis. Third, the greater muscle protein deposition observed with intermittent feeding was primarily caused by increased protein synthesis rather than reduced protein degradation. The central message is that neonatal muscle responds not only to how much nutrition is provided, but also to how that nutrition is delivered. Nutrition is often described in terms of total daily calories, grams of protein, and amounts of individual amino acids. However, metabolism is dynamic. Cells respond to changes in nutrient availability, hormonal signals, and the timing of meals. In rapidly growing neonates, intermittent feeding may create stronger anabolic signals than continuous nutrient delivery, even when total nutrient intake is identical. This does not mean that one feeding method is appropriate for every infant. Instead, the study highlights the importance of considering feeding pattern as one component of neonatal nutrition. As neonatal nutrition research progresses, the goal is not simply to deliver enough nutrients. The goal is to deliver nutrients in a way that supports their effective use for growth, tissue development, and long-term health. Today's podcast is intended for educational and informational purposes. The findings were obtained from an animal model and should not be interpreted as medical advice or as a recommendation to change an infant's feeding schedule without guidance from the appropriate healthcare team. Until next time, stay curious and keep exploring the science of metabolism.